Sushruta for NEET is now available on the Google Play Store. Try it out for free.
Be finding many more such hereditary diseases about which we have no idea at all. So, if anyone suffers from hereditary disease, is it possible to treat that disease by corrective therapy? What is called gene therapy? Yes, gene therapy is an attempt to do this. What is done in gene therapy? Now, for many of the diseases, you are very much aware that the defective genes are present on a particular chromosome. Is it? Can you tell me at any time to me? Can you tell me on which chromosome the defective gene pertaining to alpha thalassemia are present? Hello? Hello, you must answer my question.
16.
16. Yes, exactly. Alpha thalassemia related genes are present on chromosome number 16, and you also know that there are 2 genes and 4 alleles. So gene therapy, why don't you make an attempt to replace those alleles with proper alleles? That mutated allele with the proper allele. This is just I am giving an example. In fact, if at all we could succeed in making gene therapy for this alpha thalassemia, beta thalassemia, sickle cell anemia, it will be wonderful. As such, it is not done. But definitely the attempts are going on. One fine morning, we will find solution to many of our gene diseases through what is called gene therapy. Gene therapy is a collection of methods that allows correction of a gene defect that has been diagnosed in a child or embryo. When it has to be done, it has to be done in a child or it has to be done in embryo. If at all it is done in child, what will happen? If at all it is done much prior to that, what is going to happen? Let us see. So, gene therapy is a collection of methods that allows correction of a gene defect that has been diagnosed in a child or embryo. Here, genes are inserted into a person's cells and tissues to treat a disease. Here, genes are inserted into a person's cells or tissues to treat a disease. Correction of a genetic defect involves delivery of a normal gene into an individual or embryo to take over the function of and compensate for the non-functional gene. For example, we have already seen the problem with regard to type 1 diabetes. We know type 1 diabetes is due to non-production of the insulin at all. If at all we succeed in replacing this is what attempt is going on, and to a certain extent it may become successful also. So non-functional genes are replaced by functional gene. So this is what the gene therapy means. The first clinical gene therapy was given in 1990. Look here, 1990. Nearly 30 years back, first clinical gene therapy was attempted. The first clinical gene therapy was given in 1990 to a 4-year-old girl with adenosine deaminase deficiency, what we say ADA. The girl was suffering from ADA. What is that ADA? Adenosine deaminase deficiency. Why this adenosine deaminase? As you can make out, what is this? What is it? What may be adenosine deaminase means? What is it? Prithviraj? Namrata? What is it adenosine deaminase? Adenosine deaminase, which one is ending with the ACE? Which one ends with the ACE? If at all any word ends with the ACE, it is an indication that it is an enzyme. Pratiraj, it is an enzyme.
Yes, sir.
So, 4-year-old girl with adenosine deaminase deficiency means this adenosine deaminase enzyme deficiency. This enzyme is crucial for the immune system to function. Let us see in what way it is crucial. Why this adenosine deaminase is required, we shall see. The adenosine deaminase deficiency disorder is caused due to the deletion of the gene for adenosine deaminase. That means the gene which is responsible for the production of a protein which results in enzyme adenosine deaminase is deleted. The disorder is caused due to the deletion of the gene for adenosine deaminase. ADA is required for the production of B and T lymphocytes by bone marrow. So what is the function? What is the function of that? Adenosine deaminase. This particular point is not mentioned in your NCERT book. Take down. You have to take down. Navya, Rafa.
Yes, sir.
Yeah.
Yes, sir.
One thing before I forget. Those students when you collect the books from your colleges, your study material, you have to collect 2 bags. Every second year student will be collecting 2 bags. One bag will be having the study material. That means 12 books. You have 12 books for PCB, another 12 books for PCB. Study material, you will get 12 plus 12. Okay. So like that you will get mass 5 if you have opted for mass. Coming back, sorry for disturbance. So ADA is required for the production of B and T lymphocyte. B and T lymphocytes, you know, B and T lymphocytes are produced by bone marrow. So ADA is required. So when the ADA deficiency is there, these lymphocytes are not produced by bone marrow. In some children, ADA deficiency can be cured by bone marrow transplantation. That means bone marrow itself is injected, introduced, administered. In others, it can be treated by enzyme replacement therapy. Either bone marrow transplantation or enzyme replacement therapy is done in which functional ADA is given to the patient by injection. But the problem with both of these approaches that they are not completely curative. That is the problem. It is not 100% completely curative. We have to go on timely introduction bone marrow transplantation and also timely treatment with the enzyme replacement therapy. Have you understood this point? Sarvotam, Harshvardhan,
Yes, sir.
Siddesh, Charan, Vishwas. Hello? Whenever I call your name, what is that? What is that?
Yes, sir.
What is that? Indirectly taking your attendance. Understood? Sarvotam?
Yes, sir.
Okay. Now, have you understood this concept? Have you understood this concept? Savitri, Tanusri, Jayasri,
Yes, sir.
Kavita,
Yes, sir.
understood?
Yes, sir.
Any doubt?
No.
Namrata,
No doubt, sir.
Rohit, Prithviraj,
No doubt, sir.
Canis, are you there?
Yes, sir.
Have you understood Blessy,
Yes, sir.
Janani,
Yes,
Janani?
sir. Yes, sir.
Oh, so shall I go ahead, Rakesh?
Yes, sir.
Okay. So I hope it is clear to you. Now, as a first step towards gene therapy, lymphocytes from the blood of the patient are grown in a culture outside the body. Lymphocytes from the blood of the patient are grown in a culture outside the body. A functional ADA cDNA using a retroviral vector is then introduced into these lymphocytes, which are subsequently returned to the patient. ADA, a functional ADA cDNA What is that cDNA? What is that cDNA? Recombinant DNA. However, as these cells are not immortal, the patient requires periodic infusion of such genetically engineered lymphocytes. However, if the gene isolate from marrow cells producing ADA is introduced into cells at early embryonic stages, it could be a permanent cure. If at all it is done at early embryonic stage, can anyone tell me what is the difference between administering this ADA enzyme or bone marrow transplantation? In what way this bone marrow transplantation or administration of this enzyme adenosine deaminase is different in adult and in embryonic condition? Look at the last sentence. If the gene isolate from bone marrow cells producing ADA is introduced into cells at early embryonic stages, it could be a permanent cure. How you say that it is permanent cure? Are you catching my point? We are going to administer this ADA, adenosine deaminase, into a baby or into a little bit grown up, or we are going for bone marrow transplantation. In what way these two are different from isolating gene from the bone marrow cells and producing this ADA, which produces ADA, into the embryonic cells or at an early embryonic stages? Do you have any idea in what way it may be different? In what way it is going to be a permanent cure in embryonic stage and temporary cure in adult stage or baby stage? Namrata, have you understood? Jyoti, Kanisa,
Yes, sir.
come on. Who will tell me? Come on, what is that? What is that? Come on. You must know if at all any cells or any genes are introduced very early at embryonic stages, this introduction is going to have more impact because if at all you look into this embryonic development prior to 200 cell stage, you will not see any of these organs to get differentiated. You do have 200 trillion cells in your body, and these 200 trillion cells are all derived from one cell. What is that cell? What is that cell called? What is that cell called? Your 200 trillion cells of the body is derived from one cell. What is that cell called? Ayo. What is that cell called?
Zygote.
Zygote. We know that it is a zygote which is unicellular, which you can see under microscope by repeated rapid mitotic division. What do you call that? What do you call that? Repeated rapid mitotic division.
Cleavage.
What that process is called? Cleavage. By cleavage, it results in different germ layers like ectoderm, endoderm, mesoderm, and all the cells from different parts of the germ layers, they are going to produce or form different organs. So, if at all we introduce these genes isolated from marrow cells producing this ADA at a very early embryonic stage, your all cells which are going to form a particular organ will be perfectly all right, and your all cells will be having normal chromosome with a normal gene, right? Adenosine deaminase Immunodeficiency is an inherited disorder that damages the immune system and causes a severe combined immunodeficiency. You please remember SCID. Always ask a question. SCID. What is full form of SID? We also say SCID. SCID. Severe combined immunodeficiency. So it is going to affect so many different metabolistic reactions. People with this kid lack virtually all immune protection from bacteria, viruses, fungi. Therefore, it is called severe combined immunodeficiency. It causes severe combined immunodeficiency. That means those who are suffering from ADA deficiency, they'll be easily affected by either bacteria, viruses, or fungi. They are prone to repeated and persistent infections that can be very serious or life-threatening. These infections are often caused by opportunistic organisms that ordinarily do not cause illness in people with a normal immune system. If at all we have no problem with this adenosine deaminase deficiency, we will not be getting affected at all. But to those who have with the minute infection, they are going to suffer. Right? The main symptoms of ADA deficiency are pneumonia, chronic diarrhea, and widespread skin rashes. Pneumonia, chronic diarrhea, that means they affect some sort of dysentery. You know that diarrhea is a little bit mucus with a little bit blood. Chronic diarrhea means lifelong. Every day they have that problem. Widespread skin rashes. Affected children also grow much more slowly than healthy children, and some have developmental delay. Most individuals with ADA deficiency are diagnosed with the SCID in the first 6 months of life. Without a treatment, these babies usually do not survive past the age of 2. They don't survive more than 2 years. There's a problem with the ADA deficiency. In about 10% to 15% cases, onset of immune deficiency is delayed to between 6 to 24 months of age. That means 2 years now, or even until adulthood. Late onset. Sometimes it persists even in adulthood also. That is a problem. Right? This is one thing. Gene therapy, I hope it is understood. Immune deficiency in these later onset cases tend to be less severe, causing primarily recurrent upper respiratory and ear infection. Over time, affected individuals may develop chronic lung damage, malnutrition, and other health problems. All these are for your information, just to know what is this ADA. ADA. I hope it is clear to you. Now we shall move to molecular diagnosis. Right. You know that for effective treatment of a disease, early diagnosis and understanding its pathophysiology is very important. So most of the diseases are not getting diagnosed at an early stage, because at an early stage there will not be very serious symptoms. But once it reaches the advanced stage, then if at all we go to doctor, doctors say, no, now it is advanced, it is in advanced condition, we are helpless. That is what, please remember, my dear students, most of the STDs, sexually transmitted diseases, about which we have already studied, most of the sexually transmitted diseases, they are not at all getting diagnosed at early stage, but when it gets diagnosed at the later stage, it becomes very difficult to give the treatment properly to them and to bring them back to the normal condition. So, you know that for effective treatment of a disease, whatever it may be, early diagnosis and understanding its pathophysiology is very important. Please let me know from you. You must answer this question. Why that people hesitate to go to doctor at the early stage of any disease to that extent? Come on, tell me why that people don't go to doctor at early stage. Come on, quick! You have to answer my question. Have you understood my question? Many times we do not go to doctor whenever we have some problem. It is a minor problem at the beginning. Hello? Hello? Come on.
No symptoms, sir.
No, symptoms will be there. Symptoms will be there, but we don't go.
We think it's
Come on.
not a big problem.
Pardon? No. It is not that. See, many times we don't go to hospital because, come on, generally people don't like to go to hospital. Why? Why?
They'll be separated.
Huh?
They'll be separated.
Are you hearing this noise? Are you hearing my voice?
Yes, sir.
Yeah, I don't know that. I don't know what these two guys have started doing. Okay, we hope that it is not getting repeated many times. Yeah, most of the time we are fearing that even when we go to doctor, doctors may ask to get this test done, and we feel that it is some minor ailment. With a few days, it will get corrected on its own. Am I right? Am I right? Most of the time, we have health problem, we consider that these are temporary and it will get cured on its own. Like that, we leave it. And we also fear that if at all we have to go to doctor, doctors may ask to get that test, this test to be done like that. And finally, after charging several thousand rupees, then you will come to that. Yes, there is no problem at all. You are perfectly all right. This is one thing. And another thing, as you said, the symptoms may be less mild, and it may not necessitate the person, patient to go to the hospital. Right? Just a minute. Sorry for the disturbance. Okay. Just a minute. I shall just find out. 5 minutes. Okay. Sorry for the inconvenience also to you. Okay. So that is the reason. One, we are fearing that doctors may unnecessarily ask us to get a repeated number of tests to be done. Or another thing is that the symptoms are such that it is not getting warranted that you should go to the test or to the hospital. Using conventional methods of diagnosis. What is that the conventional method of diagnosis? Most of the time, whenever you go to doctor, what doctor say? Doctor will examine you, and after that, what doctors say? Come on, tell me. What the doctors say after examining you? Doctors say, doctor says, you please give your blood and let us have urine test, and sometimes in certain conditions, even feces test. But normally, you will be made to give a few drops of blood, or you will be asked to have blood test and the urine test. Using conventional methods of diagnosis, that is serum and urine analysis, early detection is not possible. Only for certain diseases it is possible, but not for all. recombinant DNA technology, polymerase chain reaction, and enzyme-linked immunosorbent assay are some of the techniques that serve the purpose of early diagnosis. So which one? Recombinant DNA technology, polymerase chain reaction, enzyme-linked immunosorbent assay, ELISA. Out of these three, you already know how many? What all you know? What all you know? You know recombinant DNA technology and polymerase chain reaction. For which one this polymerase chain reaction is done recently?
COVID.
COVID test, where our DNA is made to get multiplied in a number of copies to test it. So polymerase chain reaction requires this RNA recombinant DNA technology in many cases. And enzyme-linked immunosorbent assay are some of the techniques that serve the purpose of early diagnosis. Okay, these three you have to remember. Presence of a pathogen, bacteria, virus, etc., is normally suspected only when the pathogen has produced a disease symptom. If at all we have fever, that is a symptom. If at all you have headache, that is a symptom. If at all you have body ache, stomach ache, that are symptoms. Presence of a pathogen, especially bacteria, virus, is normally suspected only when the pathogen has produced a disease symptom. If the symptom is not seen, as you find sometimes, now, symptoms are not seen. It is already discussed in many of these nowadays for the last 2 years. What is done? You may be, it is said, you may be corona positive without a symptom. Have you heard about that? Have you heard about that? Yes? Without a symptom, one may be corona positive. Symptoms may not be seen because of the resistance power of that particular person. And what I doubt, what I suspect is that mostly all of us, we have got infected by this COVID-19, but the symptoms did not appear in us because of our strong resistance power. Okay, but sometimes what happens, there will be presence of pathogen, especially bacteria viruses, is normally suspected only when the pathogen has produced a disease symptom. Without a disease symptom also, sometimes pathogen may remain inside our body. By this time, the concentration of pathogen is already very high in the body. However, very low concentration by bacteria or virus at a time when the symptoms of the disease are not yet visible can be detected by amplification of their nucleic acid by PCR. That is why what test is done? What test is done? For COVID-19, what test is done?
PCR test.
PCR test. Yes? So, this is just to amplify their nucleic acid. Right? PCR is now routinely used to detect HIV in suspected AIDS patient. We can also add to this. PCR is now routinely used to detect COVID-19 cases in suspected patients. It is being used to detect mutations in genes in suspected cancer patients also. It is a powerful technique to identify many other genetic disorders. What is polymerase chain reaction? A single-stranded DNA or RNA tagged with a radioactive molecule, some radioactive molecule is tagged to that as a probe, is allowed to hybridize to its complementary DNA in a clone of cells, followed by detection using autoradiography. So, what is done? A single strand of DNA or RNA, which is tagged with radioactive molecule, that is what is required. We have to see where this radioactive molecule easily you can observe that is allowed to hybridize, go on multiplying later to its complementary DNA in a clone of cells, followed by detection using autoradiography. The clone having the mutated gene will hence not appear on the photographic film because the probe will not have complementarity with the mutated gene. The clone having the mutated gene will hence not appear on the photographic film because there is no complementarity with the mutated gene, because the probe will not have. What is that probe? Probe is a radioactive molecule, right? So this is what you see here. ELISA, we have just said about ELISA, another test. ELISA is based on the principle of antigen-antibody reaction. This is only one sentence you have about this ELISA test. It is always based on the principle of antigen-antibody interaction. Whenever you come across antigen-antibody complex, it enables us to know that yes, this particular pathogen or this particular disease is there. Infection by pathogen can be detected by the presence of antigens, proteins, glycoproteins also, or by detecting the antibodies synthesized against the pathogen. So, whenever some pathogen attack our body produces a large number of antibodies. By blood test, we can find out, yes, this is the problem with regard to this, because these antibodies are produced in the body. That means this pathogen has entered the body. So, many times, which particular pathogen the disease-causing microbe has entered into the body is detected on the basis of a blood test, which indicate the presence of these antibodies. Now, let us look into one more, transgenic animals. You might have heard about many transgenic animals. What are these transgenic animals? Animals that have had their DNA manipulated to possess and express an extra gene are known as transgenic animals. Animals with the extra genes in their body, foreign gene in their body, they are called transgenic animals. What has happened? Some DNA is introduced into the body of these transgenic animals. Transgenic rats, rabbits, pigs, sheep, cow, and fish have been produced, although over 90% of all existing transgenic animals are mice. Remember, even though we have been successful in getting all these transgenic animals, what are these transgenic animals? These are the transgenic animals and animals to whose body we could successfully introduce this extra or foreign gene. So even though we have transgenic animals on these different categories, most of the transgenic animals which are extensively used in the genetic experiments are mice. Normal physiology and development. Transgenic animals can be specifically designed to allow the study of how genes are regulated and how they affect the normal function of the body and its development. That means, if at all we know that these genes, one particular gene, let us say that A gene. A gene is responsible for X disease. Now, what are you doing? We are just taking this A gene and starting putting into the different animals and trying to find out what these genes do at the different stages of the life cycle of the animal. So, transgenic animals can be specifically designed to allow the study of how genes are regulated and how they affect the normal function of the body and its development. Example, study of complex factors involved in growth, such as insulin-like growth factor. If at all we want to know how that insulin is responsible for the metabolism, sugar metabolism, we are just looking into that by just introducing some of this that gene, as we said here, study of complex factors involved in growth, such as insulin-like growth factor. By introducing genes from other species that alter the formation of this factor and studying the biological effects that result, information is obtained about the biological role of the factor in the body. That means one gene, introducing one gene and studying it in mice, what this particular gene does in mice and what effect it is bringing about. So, mice, monkeys, rabbits, horses, horses, these are all extensively used before any treatment or before any use of a drug is made in human body. So, it will, because all of us we are mammals. So, this enables us to know how a particular gene works and how it affects the normal metabolic activities. Study of disease. Many transgenic animals are designed to increase our understanding of how genes contribute to the development of disease. As I told you, we have a number of diseases which are genetic, which are gene regulated. These are specially made to serve as models for human diseases so that investigation of new treatments for disease is made possible. Today, transgenic models exist for many human diseases such as cancer, cystic fibrosis, rheumatoid arthritis, and Alzheimer's disease. Many transgenic models exist, but still today we have not got a conclusive result. If once conclusive results are available, it will be definitely incorporated into in our society for the welfare of human beings. What do you mean by conclusive result? Please remember, if at all we just try with one particular gene, and we wanted to see that how this gene is affecting or successful in treating a particular disease like cancer, what do you want? This particular transgenic model where this treatment to the cancer is tried. If the same gene we are trying with the human being, it should bring about 100% cure, not 99.99. I hope before this vaccination was tried with regard to corona, there is a lot of hue and cry. You know what is that? Do you know what is that hue and cry was there? People were opposing to get this vaccine administered to their body under one pretext. Can anyone tell me what is that? What is that? You know, now all the people have gone for vaccination. But prior to administering this vaccination, people used to make, especially opposition parties and all, they used to say, no, we don't want to take this Indian vaccine under the pretext because they are, what is that? Very inverted commas. Anyone tell me? Rakesh, Pratyuraj, Siddesh, Harshvardhan, or
Not safe, sir.
no, not that. It is not that. It is not that. There is another term. I wanted to you to tell me. Kanisa, Navya, Aisha, Rafa, Blessy, Diti. That is, all used to say it was not properly got clinical trials. Have you heard about that? Have you heard about that word? No? Yes or no? Clinical trials. That means before administering any vaccine to the society, any vaccine in society, it should undergo sufficient clinical trials. Means if at all it is tested with 100 people, and in all 100 people, it should give the same result without any bias. So, therefore, many times, even though some of these that cure for these diseases are trying, yet to get a 100% clearance. So these are all are tried. Not these models. I'm not telling it. I don't know at what stage, whether it is in a mice stage or rabbit stage or monkey stage. There are different stages they try. So it must be tested in a different types of animals. Then it must be tested in human beings in a different places, and then the finally a 100% when they convince that it is giving 100% cure, then only it will be approved by the Indian Medical Council. Now coming to biological products. Medicines required to treat certain human diseases can contain biological products, but such products are often expensive to make. What do you mean by these biological products? Transgenic that produce useful biological products can be created by the introduction of the portion of DNA or genes which codes for a particular product such as human protein, alpha 1 antitrypsin used to treat emphysema. So, alpha 1 antitrypsin deficiency is one of the reasons for emphysema. Have you heard about emphysema? Have you heard about emphysema? Hello? Anyone knows about emphysema?
Yes, sir.
What is it, emphysema?
So the lungs is affected.
Yeah, very good. Lungs are affected, and lungs alveoli of the lungs lose their ability of exchange of gases. They will not allow exchange of gases to take place properly. They become defunct. So we have certain product such as human protein, alpha-1 antitrypsin is used to treat emphysema. So similar attempts are being made for treatment of phenylketonuria and cystic fibrosis. Right? So this is biological product. Transgenes that produce useful biological product. This is what biological product that we have just to treat one or the other diseases. Similar attempts are being made for treatment of phenylketonuria. I hope you know what is PKO. You recall that? Who will recall? Come on.
Inborn error of metabolism.
What happens?
The enzyme phenylalanine is not converted
Phenylalanine?
to tyrosine.
Yeah. So that what will happen? It gets accumulated. In what form?
Phenylpyruvic acid.
Phenylphenic acid. And ultimately it results in what?
Mental retardation.
Mental retardation. And what is cystic fibrosis? Yes? Have you heard about cystic fibrosis? Anyone?
Mucus filled in the lungs.
Not only lungs, in air passages, and so that the air movement in the bronchial tube gets affected. So this is what emphysema. Emphysema is called an obstructive lung disease. This is alveoli with emphysema. These are the normal alveoli. Emphysema is called an obstructive lung disease because the destruction of lung tissue around the smaller sac called alveoli makes these air sacs unable to hold their functional shape upon exhalation. Thereby, they affect the exchange of gases. Right? And this is what you have. You have heard about phenylketonuria, just for your information. Again, I'm bringing it to just remind you or recall that is another disorder that occurs approximately 1 in 5,000 to 6,000 births. Remember, 1 in 5,000 to 6,000 births. Why Tanasi? Hello? ನಾನು ಕ್ಲಾಸಲ್ಲಿದ್ದೇನೆ ಮತ್ತೆ ಮಾತಾಡಲಾ Okay, so this phenylalanine is 1 in 5,000 to 6,000 births. It is not a small thing. 1 in 5,000 to 6,000 births suffer from PKU. The condition is due to a missing enzyme that metabolizes an amino acid called phenylalanine, which the body requires in order to function normally. Phenylalanine is a precursor to tyrosine, the monoamine, signal molecules of dopamine, norepinephrine, epinephrine, and the skin pigment melanin. For all these, tyrosine is required. Tyrosine is only produced when phenylalanine is metabolized by the enzyme phenylalanine hydrogenase. So that is not getting produced properly. Because individuals with PKO lack this enzyme, there is an accumulation of phenylalanine in blood and a related substance in urine, phenylpyruvic acid in blood and a related substance in urine. This is the condition of PKU. The patient suffers from seizure, epilepsy, or fits. I hope you might have heard about. Have you heard about epilepsy or fits? The person
Yes.
all of a sudden loses consciousness. He starts jerking movement. That is what is called seizures. Profusely salivates. Starts rolling down on the ground. Right? For about few minutes, he will be doing like that. Later, after about 5 minutes to 10 minutes, and even he just moves that straightens the limbs and all. And later, all of a sudden, he wakes up and looks around, comes to normal. But that what he has been done for that last 10 minutes, he will not absolutely, he will not be knowing anything. During this seizure or epilepsy, thousands of neurons get lost. Have you heard about this? Have you seen the patient suffering from this? Any one of you? If so, what people do all around the world, not only in India, if anyone suffers from epilepsy, immediately they do something. Hello? They do something to the patient.
They will give metal object in their hands.
Yeah, generally they give a key bunch or some other metal in the hand. I don't know the effectiveness of that, but it is done throughout. So that is what. And you also know these people who suffer from seizures or epilepsy or fits like that, under two circumstances, their condition gets worsened. The moment if they see fire, they will jump to the fire. The moment they see water, they jump to the water. They jump to the water. And in one of the known cases to me, a person who used to carve with his nail human portrait, who used to do that human portrait, if at all you go and sit, or if you go give him one photo, yet he takes some wedding card, type of a card, and he just with the nail, he used to just carve the picture of the person, portrait, and is so famous with his that art, and he got appreciation letter from none other than our late Prime Minister Indira Gandhi. He got personal letter from her appreciating his skill. That person was suffering from epilepsy, and one day he got epilepsy when he was near the well, and he fell down to the well and lost his life. Okay, so this PKU, because individuals with PKU, they lack this enzyme, they suffer from many of the conditions like patients suffer from seizure, tremor or trembling and shaking, stunted growth, hyperactivity, skin conditions such as eczema. Have you heard about eczema? A musty odor of their breath, skin, or urine. Have you heard about eczema? Have you seen patients with eczema? Eczema patients. This is what seizures. Seizures occur when there is an abnormal surge of electrical activity in the brain due to chemical changes in nerve cells. Typically, there is a balance between excitatory and inhibitory brain cells. Excitatory brain cells cause activity and inhibitory brain cells prevent. Seizures can occur when this balance is disrupted and there is either too much or too little activity. This is what's happening. Gradually, they lose more and more brain cells. And that loss is lost forever. Right? This is eczema. Right? So this is due to one of the reason for due to is the PKU. This is cystic fibrosis, accumulation of the mucus. This is normal bronchiole or active. This is the infected one. This much whatever that is there, it cannot be removed. Absorb at all because of the accumulation in the intestine. Rosy produces human protein enriched milk. Human protein enriched milk. Right? How much? 2.4 grams per liter protein contained. The milk contained the human alpha-lactalbumin and was nutritionally a more balanced product for human babies than natural cow milk. So they could get this transgenic cow. This cow and all, even if at all you develop through this transgenic way by administering this recombinant DNA technology, in the nature they have to fight with the environmental conditions. That becomes difficult. That is why most of the time we are going to this normal method of this breeding. This is with regard to, just remember, transgenic rosy. Now let us look at the vaccine safety. Transgenic mice are being developed for use in testing the safety of vaccines before they are used on humans. I told you, any vaccine, any injection, any drug, if at all it has to be used by human being, if at all it has to come to the market, if at all one has to get a permission for the production of that particular medicine, it has to be tested in a number of cases. Transgenic mice are being developed for use in testing the safety of vaccines before they are used on humans. Transgenic mice are being used to test the safety of the polio vaccine. If successful and found to be reliable, they could replace the use of monkeys to test the safety of batches of the vaccine. Many times, mice, in some cases, rabbits are used. For all this, you can grow, you can rear a rabbit. You might heard about there are wild rabbits, and also you do have the white rabbits, albino. I have reared these albino rabbits, and within 3 months you'll get to the progeny, right? I have tried. So coming back here, these particular different animals are tried for this before administering that. One more thing I wanted to tell you. Even before any particular medicine is manufactured on a large scale, before it is permitted to be sold in the pharmaceutical shops, this medicine has to be tested in a number of human beings secretly. When I was working for KMC Manipal, when I was engaging classes for them, I had some contact with some doctor. To them, I came to know many of these new tablets, whatever they wanted to be brought to the market, these some of the tablets before getting branded, before getting final permission, they are secretly tested on human beings. How? There are some people who come forward for that, and there is the earning for them. That's earning for them. Now let us see that one new tablet they wanted to administer with regard to your headache. This particular tablet is not there in the market. Are you hearing me? Hello? Are you hearing me?
Yes, sir.
Yeah. This new tablet, so far is not available at all. Or let me just tell you, at present, we don't have any tablet, just like a family planning, what a tablet is used by women. What is the tablet used by women? What is it simply called? Family planning tablet. What is it simply called?
Contraceptive.
Mala D.
Mala D. Rakesh, you remember Mala? Okay. So this, or simply it is called Mala D. Rakesh. It is a brand. What is it called otherwise? Oral, oral, oral pills. Oral pills. Oral pills. Oral pills are there only for women. Suppose we want to develop some oral pills for men. Let us say that if at all these oral pills are taken by men, Even though he produces a sperm, let us say that it is going to make the semen with the defective sperms or with the, let us say that, decrease in the sperm count. So this oral pill, if at all we, this is hypothetical, 100%. These oral pills we first try with the mice. We try the rabbits. We try the monkeys. Now we want to try with the human beings. Is it possible that much easier to try with human beings? For that purpose, some people will come forward. Yes, you test that with me. I am ready to undergo that test. After making some individual human beings get tested, they have to give the report, that the doctors have to give the report to the medical companies. But the thing is that the people should be ready for that. Or this is, of course, it is optional. One can go for that, one need not to go for that. But if at all you take a headache, the people who are coming with headache only, that you can't get headache whenever you want, those people who are specially recognized for that purpose, meant for that purpose, every hospital will be having a couple of such people with them. They always be ready for this purpose. Suppose that whenever they get headache, they come to the doctor, or doctors will keep them informed. Whenever you get a next time headache, please come to me. Whenever you get a next time stomachache, please come to me. They'll try and they'll see what is the result in them. In that way, after testing repeatedly, then that particular knowledge will be used in another couple of people with under full guidance, under full observation. So this is what vaccine safety. If successful and found to be reliable, they could replace the use of monkeys to test the safety of batches of the vaccine. Like that, it may be tested. It is for the safety of batches in some other human beings. Chemical safety testing. This is known as toxicity or safety testing. The procedure is the same as that used for testing toxicity of drugs. Transgenic animals are made that carry genes which make them more sensitive to toxic substances than non-transgenic animals. Transgenic animals are made that carry genes which make them more sensitive to toxic substances than non-transgenic animals. They are then exposed to toxic substances and effects studied. How much a toxic substance gets accumulated in body, and what will be the response there? Many times, whenever you are administered with antibiotics, doctor will ask you one question. You know what is that? Hello? Before administering any antibiotics, generally doctor will ask one question. What is that? Hello? No one knows? Before, hello, Caris, are you there?
Yes, sir.
Whether doctors ask any question, do you know? Prithvi? Rakesh? Generally, before administering any antibiotics, a doctor has to ask the patient, have you taken antibiotics before? Are you sensitive to antibiotics? If at all you say that, yes, I have taken before, immediately they administer the antibiotics. Otherwise, they have to find out how much sensitive you are to that by administering mild dose. Transgenic animals are made that carry genes which make them more sensitive to toxic substances than non-transgenic animals. They are then exposed to the toxic substances and the effects studied. Toxicity testing in such animals will allow us to obtain result in a less time. Ethical issues. The manipulation of a living organism by the human race cannot go on any further without regulation. Manipulation of living organisms by the human race cannot go on any further without regulation. Now, can we get transgenic animals for all our need? For all our need, can we have that transgenic animal? Or putting in another way, you know that now we have become very successful in cloning. We have become very successful in getting robots. Am I right? Do you know that? At some restaurant, robots only come and take order and deliver the eatables to you. Have you heard about that? Have you heard about that?
Yes, sir.
Have you seen? Have you met any robots? Have you seen any robots welcoming you to the shop, taking orders from you, delivering the goods for you? So, in this way, there are some ethical issues with regard to this transgenic animals also. The manipulation of living organisms by the human race cannot go on further without regulation. Some ethical standards are required to evaluate the morality of all human activities that might help or harm living organisms. Going beyond the morality of such issues, the biological significance of such things is also important. Genetic modification of organisms can have unpredictable results when such organisms are introduced into the ecosystem. Right? Now, if at all we are in a position to prepare human robots, why don't you get more and more robots and use it for each and everything? Come on, come on. Now you know robots can cook food for you. Robot can do your all house chorus work, but there's a regulation that even though we are in a position to do that, there's a regulation because we are suspecting some unpredictable result may happen in future. Therefore, the Indian government has a set of organizations such as a GEAC, Genetic Engineering Approval Committee, which will make decisions regarding the validity of GM research and the safety of introducing GM organisms for public services. This is what we are not at all permitted to introduce whatever GM organism into society for public services. The modification or usage of living organisms for public services as a food and medicine sources, for example, has also created problems with the patents granted for the same. Many times, patents are granted, and we are finding now the problem regarding patent. I shall tell you about some of the patent which you may be knowing, and for which Indian government has to fight with the fight in the international court. There is growing public anger that certain companies are being granted patent for products and technologies that make use of the genetic material, plants, and other biological resources that have long been identified, developed, and used by farmers and indigenous people of a specific region or country. You know what is this? What is the meaning of this? Can anyone give me the example for this? There is growing public anger. By whom? By the farmers, by the villagers, that certain companies are being granted patent for products and technologies that make use of the genetic material or plants and other biological resources that have long been identified, developed, and used by farmers and indigenous people of a specific region or country. Jyoti, Jyoti, Jyoti,
Yes, sir.
are you going to kitchen
Yes, sir.
very,
I go to kitchen.
very often?
Sir?
If at all, while chopping this vegetable, okay, or chili, if at all your mother gets her finger cut slightly by the knife, what mother does?
Sir, she washes her hand and applies turmeric.
Apply
Turmeric?
turmeric powder. Am I right? Turmeric powder.
Yes, sir.
Yeah. Now, if at all I say you are using turmeric powder. How your mother knows that she has to apply turmeric powder? How you know? How your mother knows? Did you at any time ask her?
No, sir. She knows it's like a medicine. That it acts like a medicine. That's
Oh
right.
my goodness. Then you are not the right choice for my question. Can anyone tell me how that mothers know about that? How mothers in kitchen know that if at all their finger get cut, immediately they have to wash and use turmeric? How they know? Anyone? Boys or girls? Boys or girls, please answer me. How they know? Quick, 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 quick. Hello? Hello?
Sir,
Number
maybe
of...
their parents would have told them.
That is true. Their parents knew. And their parents, your mother knew, because your mother's mother knew, because your mother's grandmother knew, because your mother's grandmother knew. If at all tomorrow someone comes and tells, and this is what we have to do, if at all we get cut our fingers, we have to use turmeric, and this is what I have found out, and it is my patent. If at all I am given that patent, if at all I claim that I am publishing that and I am telling that you have to use, you can use the turmeric as antimicrobial to get your fingers protected from microbes. And I want to get a patent for that. Do you agree? Do you agree that I may be given patent? Do you agree? Do you agree? After I get patent, tomorrow if at all you want to use the turmeric, you must get permission from me. You must give some fees to me for this. Because I'm telling today that the turmeric is an antimicrobial, it has antimicrobial property and you can use it. This is what is happening. Can you buy this? Can you just tell me what all you do like that? Do you know? India has to fight against that. Are you understand these sentences? Now, if you look here, rice is an important food grain, the presence of which goes back thousands of years in Asia's agriculture history. There are an estimated 2 lakh varieties of rice in India alone. The diversity of rice in India is one of the richest in the world. 2 lakh varieties of rice in India. Basmati rice is distinct for its unique aroma and flavor, and 27 documented varieties of basmati are grown in India. You ask your grandpa, grandma, grandpa's grandma, grandmother's grandma also knew about basmati rice. Okay? Why you use it? Why you use that basmati rice? Are you using this basmati rice for everyday eating? Lunch, breakfast, lunch and dinner? Or you use it for a specific purpose? Come on. Why you are using basmati rice? Come on. Have you seen basmati rice? Kanisa? Aisha?
Yes, sir.
Why you use
Yes,
it?
sir.
Tanusri?
Biryani.
Savitri? To make?
Yes, sir.
What?
Biryani.
Huh?
Biryani.
Biryani. Okay. There is a reference to basmati in ancient text, folklore and poetry, as it has been grown for centuries. Now, in 1997, an American company got patent rights on basmati rice through the US Patent and Trademark Office. After that, what happens? If anyone wants to grow basmati rice, you should get the permission and should to pay for that. This allowed the company to sell a new variety of basmati in the US and abroad. This new variety of basmati had actually been derived from Indian farmers' varieties. Only because of our basmati varieties, they just developed it and they're telling that we got patent and you should give charges for us. Indian basmati was crossed with the semi-dark varieties and claimed as an invention or a novelty. Do you agree with regard to this? Do you agree that basmati patent can be given to American company? When you are using this, since the time immemorial, are you ready to give to allow the company to get the patent and tomorrow you are ready to pay for that?
No,
Tell
sir.
me. No,
No.
the patent extends to function equivalents, implying that other people selling basmati rice could be restricted by the patent. So, several attempts have also been made to patent users, products, and processes based on Indian traditional herbal medicine, turmeric and neem. You know, people use neem right from morning in day-to-day life. Take a small stick of neem and use it as a brush. We are using neem for each and everything. If we are not vigilant and we do not immediately counter this patent application, other countries or individuals may encash on our rich legacy and we may not be able to do anything about it. Can we give it up that patent right to all other people from all other nook and corner of the world? Our product for whatever that we are using since the time immemorial in our day-to-day life? These are the things. Understood? So, biopiracy is a term used to refer to the use of bioresources by multinational companies and other organizations without proper authorization from the countries and people concerned without compensatory payment. If at all, some people are making use of our product, our knowledge, and if at all they want to make money out of that, it is called biopiracy. It is the stealing our knowledge. It is the stealing of our material. Many times, suppose there are, let us say that there are about 50 species of medicinal plants only available in India, in Western Ghat. And if these 50 medicinal plants, 50 medicinal plants are systematically, they are just exported to some foreign country. And tomorrow, if the foreign country says that these medicinal plants are ours, and you should pay for that because they do some hybridization and they start cultivating that, but originally it is from our place. So biopiracy is the term. Always ask a question. Biopiracy is the term used to refer to the use of bioresources by multinational companies. They are making stealing and other organizations without proper authorization from the countries. It is a medicine plants. 50 medicine plants are ours. We have grown. We are taking care. It is our biological wealth. If some other person tomorrow claims that it is theirs, it is not proper. If they want to claim that they want the patent for that, they must pay us for the claiming their patent. I hope you are understanding my point, right? Most of the industrial nations are rich financially, but poor in biodiversity and traditional knowledge. Unfortunately, we are rich in biodiversity and traditional knowledge, but poor in industrial products. In contrast to the developing and underdeveloped world is rich in biodiversity and traditional knowledge related to bioresources. Traditional knowledge related to bioresources can be exploited to develop modern applications and can also be used to save time, effort, and expenditure during their commercialization. There has been growing realization of the injustice, inadequate compensation, and benefit sharing between the developed and developing countries. Many times, it is our product, it is our natural wealth, but other countries are making are exploiting our natural wealth, or exploiting our knowledge, and trying to make earn money by claiming patent. Therefore, some nations are developing laws to prevent such unauthorized exploitation of their bioresources and traditional knowledge. The Indian Parliament has recently cleared the Second Amendment of the Indian Patent Bill that takes such issues into consideration, including patent terms, emergency provisions, and research and development initiatives. Right? Clear? So, do you have any doubts on this? Hello? Hello? Okay. So, with this particular chapter got over.